For the next presentation is anti-tumor activity of dual alpha B7, H3 alpha CD3, and alpha PD-L1 alpha CD28 bispecific T-cell engager arm T cells against prostate cancer by Yanapith Teerachattrawat from Faculty of Medicine, Siriraj Hospital, Mahidol University.
Greetings, my name is Yanapith Teerachattrawat, a third-year medical student from Siriraj Faculty of Medicine, and now I'm going to present this project, an anti-tumor activity of dual anti-B7H3 linked to anti-CD3 and anti-PD-L1 linked to anti-CD28 bispecific protein engager armored T-cell or BAT against prostate cancer. Here's the outline. We got introduction, objective, methodology and result, research plan, and lastly, bibliography. Start with the introduction. Prostate cancer is a common tumor found in male with the second most diagnosis globally. And here's standard treatment. We got like hormonal therapy, radiotherapy, or surgery. But this conventional treatment consists of like limited precision and effectiveness and excessive side effect. Moreover, we're concerned about the metastatic castration-resistant prostate cancer, or mCRPC. It's a type of aggressive prostate cancer, which is like, it's resistant to hormonal therapy, and it's risk of relapse and metastasis. So what we interest is immunotherapy. Current immunotherapy in prostate cancer, we got like monoclonal antibody, and in process of development like CAR T cell. CAR T cell is like modified T cell of the patient, and it's costly in terms of production, and moreover, it has an antigen escape risk. And here we got like bispecific antibody and protein engager, but its downside is it has a short half-life, so we need to use the continuous dose with the patient, and it's risk of cytokine storm syndrome. So what we intend to do is we use this duo. We have a bipe. It's like a bridge between T cell and tumor cell, and we arm them with the T cell before like give back into the patient. And this bipe, it targets the B7H3 and PD-L1, which express in like prostate cancer is immunocheckpoint. So we propose that this dual anti-B7H3 linked to anti-CD3 and anti-PD-L1 linked to anti-CD28 dual BAT can enhance the effectiveness of anti-tumor ability and T-cell proliferation. Here is the objective. Is to develop and evaluate the anti-tumor ability and safety of dual BAT against prostate cancer. And here's the methodology. So the methodology and the result, we start with, we need to check the expression of B7H3 and PD-L1 in prostate cancer cell line. We got 4 cell lines, DU145, IOCAP P1, IOCAP wild type, and VCAP. And first, we use flow cytometry technique. And the graph in the right, it can show you that B7H3 is highly expressed in LNCAP P1 and LNCAP wild type. Meanwhile, PD-L1 is properly expressed in DU145. Moreover, we use the technique like immunofluorescence. We show the expression in green color and red color here. And we use the immunobot analysis technique to validate the expression of them. And the result is consistent with the first part, that LNCAP P1 expressed highest in B7H3, and the U145 is expressed highest in PD-L1. Here's the part of preparing the bipe. Actually, we got bipe scaling stable cell from Dr. Kwan Pimon. And here is the structure of these 2 bipe. And we need to validate bipe via immunobot analysis. Next, we need to evaluate binding ability of anti-B7H3 linked to anti-CD3 and anti-PD-L1 linked to anti-CD28 bipe. We use anti-B7H3 with LNCAP P1 and anti-PD-L1 with U145 since it's highest expressed in the first step. And the graph shows that the percent binding is correlated with the antigen expression and by concentration. Next, we need to evaluate the binding ability of anti-B7H3 linked to anti-CD3 and anti-PD-L1 linked to anti-CD28 bipe with 2 parts, T cell and with our target cell. So with T cell part, we have an expression of the donor here is CD3 and CD28. And graph in the right also show that the binding correlates with the BiPE concentration. And we got the optimized BiPE concentration that we can use in the next arming step. Next, we generate the dual BAT. And we check the cytotoxic ability in 2D models. Here is a picture of dual BAT and prostate cancer under the confocal microscope. The green one is T cell that armed with BiPE, and the pink one is prostate cancer, and it attached to the BAT attached to prostate cancer here. And the graph in the right is a graph from X-Celligent technology. It's like a technology that observes the cellular adhesion of the cell. And we treat that cell, the prostate cancer cell line, DU145, with dual BAT. And dual BAT demonstrates the greatest reduction of the DU145 cell index in 2D cytotoxic assay. And we plan to do in ANCAP P1 in the next part. Here's conclusion that we got. We got that BCL history was highly expressed in prostate cancer cell line, while PD-L1 was upregulated in some. And we got that anti-BCL linked to anti-CD3 and anti-PD-L1 linked to anti-CD28 bispecific protein engager or BiPE generated from stable cell line could bind to target prostate cancer cell and effector T cell. And we got that dual BAT demonstrated the greatest reduction in the DU145 cell index in 2D cytotoxicity, exceeding the single arm one. In short, dual BAT are in the process of evaluating their cytotoxic function, proliferation, and cytokine secretion. Here's the acknowledgement. Here's the bibliography. Thank you for your kind attention.
So after the BiPE attached to your target cell, the proliferation is stopped or just remain? What happened later?
From the literature review, it said that BiPE can enhance the T cell proliferation, but we did not in the process of checking that part yet. But we armed it with T cell and before put back into patient, so we intend that it might be like help in T cell proliferation before.
Here you have showed a lot of information about the efficacies of your new dual BAT. What about the specificities of these molecules?
You mean the specificity of Bispecific?
Of the dual BAT to the cancer cell. So
Oh.
if you expose dual BAT with the low antigen, for example, cell, how is gonna work?
In PD-L1, it's quite low, as you can see. But PD-L1 is like progress with the tumor step. Like tumor in late state have a highly expressed of PD-L1, especially in prostate cancer. And actually, it's immunotherapy, so precision is needed. And I think in the term of treating real patient, we need to biopsy that tumor and see the expression before treating some kind of this immunotherapy. Yeah.
Can you test it in vitro first for the specificities, and can you suggest the approach to test the specificity of your dual BAT here?
In vitro?
Yes.
Here we test with the cell line, right? And the cell line of DU145 is expressed highest in PD-L1, so it might work well. And we have LNCAP that has expressed highest expression of BCL-HT. So we intend to treat with two of them. So and we so that's how we treat in vitro. But in vivo is the next step if it works. Yeah.
What about the half-life of the dual BAT? Is it better compared to bispecific antibody?
we hope that it's better than only protein engager because if the BiPE one, we just use protein engager into like patient, but here we excrete the blood, we got T cell, and we arm first, and we hope that it might work better than BiPE because BiPE has like more short life. And actually, in our laboratory, we have tried this bat, and the result of is hopeful than BiPE one. Yeah. Thank you, everyone.